I'm giving the first session on speed breeding, and it's going to be a speed talk as well. So considering we've only got about half an hour left, but I think I can go through it all. And so I'll talk mainly about the technology itself in terms of rapid generation advance. On Friday, Cecile Richard will talk more about the methodologies that we're adopting in the system for selecting for traits. So biotic and abiotic traits. That's the exciting space as we start to combine this sort of technology with other tools. So I guess I should introduce myself. I'm Dr. Lee Hickey from the University of Queensland, and so there I lead a group working on wheat and barley genetics. And so this is some of my team. We refer to ourselves as the Hickey Lab, and we're doing a lot of things on social media. So you might, those of you who are on social media, I'm already connecting with some of you, but if you are, please get in touch so we can share things and communicate. So. Everyone here is at this workshop because they understand that we need these technologies to accelerate genetic gain for wheat breeding. It's a slow and painful process. Even using the cutting edge approaches today, it's still slow, and there's opportunities to speed that up. The big companies are refining things, but still, I think there's advances to be made there as well. So it's all part of our goal is to reduce these breeding cycles, because it can take up to 20 years to develop a variety. It's a sort of a throwaway number, but it all depends on whether you're making an elite by elite cross or you're trying to introduce novel alleles into an adapted variety for farmers to grow. It can take several breeding cycles. And so the key part of that is to reduce the breeding cycle and speeding up this generation advance. And two key ways that people have done this in the past through shuttle breeding, so developed by the famous Dr. Norman Borlaug back in the day. And of course, a double haploid technology, probably two of the most renowned activities for speeding up generation advance. So most of you are probably familiar with shuttle breeding. It was basically developed by CIMMYT, by Dr. Norman Borlaug, shuttling the germplasm from the north of Mexico to the south, so from Obregon down to Toluca and El Batan. And by doing this, because the conditions were quite different, they could achieve 2 generations of wheat in a year. And so cutting the time required for breeding new varieties essentially, or theoretically, in half. So rather than growing just 1 generation per year, you grow 2, you're going twice as quick. So that's cool. So shuttle breeding, I guess, has been extended and adopted not only in wheat, but in other crops. So we think about maize. They have summer nurseries, winter nurseries. It's a summer crop. So winter nurseries in Hawaii, or they'll have other nurseries in Chile. And so shuttling germplasm around the world is a great way to achieve more field environments. And we heard a little bit about CIMMYT and how they've adopted this and extended it to incorporate better resistance to UG99 in Africa. So they had this sort of 3-way shuttle breeding sort of system operating. Which was really aimed at tackling broadly adapted material, but also incorporating good rust resistance. So double haploid populations, I guess you can see there, it's an example of if you were to do make a cross and go through the process of self-fertilization to fixation, and you did that 1 generation in the field at a time, and you only have 1 growing season, it takes many years. Although companies in developed countries are actually speeding this process up a lot, there's a lot of gain to be made in developing world countries where they often only achieve one generation per year, and depending on the crop, of course. So we're talking about wheat here today, but a lot of other crops are pretty slow at doing this as well. So in comparison, double haploid technology can take anywhere between a year or two. So the process is quite fast, but still it takes time. Unless you're doing it in-house, you have to send the seed to the company, and then by the time they go through this process, they send it back. You get a small number of very shriveled, crappy grain, and then you have to increase it. So it's another generation before you can actually do anything with it, and then probably you still don't have enough to actually do proper field trials or anything like that for another generation. So yeah, I mean that's part of the problem. So when we weigh these things up, the DH technology, I guess, has been a lot of good aspects, and it's been a revolution in terms of helping us speed up that process to fixation for mapping studies. These populations give us great power to detect QTL because you get these big chunks of DNA staying together, give us great power to identify QTL. But there's a lot of, and of course it's completely fixed, well at least for a generation or two until you get mutations, and of course that just happens. But the disadvantages I obviously want to highlight here. I'm talking about speed breeding technology and so maybe some advantages of that, but obviously you can't perform early generation selection in double haploid technology. You get what you get, so it's a random combination of genotypes. You get less recombination, and it's obviously very laborious, and you need specialized skills. It can be expensive. So I had a chat with Hans in the gym the other day. We were doing a workout together, pumping some weights. No, I'm just joking. But I had a chat with him in one of the meeting rooms, and he said he was concerned about people wanting him to use double haploid technology and to scale that up. But when you think about it, what's the cost and the effort required to do 100,000 double haploid lines, it can get pretty pricey. So I guess some of the downsides. But so the speed breeding innovation, I guess, was for us at UQ was born about 8 years ago when we heard about NASA's crazy ideas about growing wheat in space to provide a food source on long space or shuttle missions. So the way that they were doing it was they were using constant light and temperature or control temperature to accelerate plant development. And they achieved very rapid plant growth. And so we thought, well, what a great tool it could be to accelerate our research. And so we started playing with it at UQ and with myself and a couple of others, including Ian DeLacy and Mark Dieters. And when we first started, we got very poor quality plants. So it wasn't so easy. The plants were growing so fast. We had a lot of problems. They weren't taking up nutrients very well, etc. So over the last 8 years or so, we've refined the system and worked out a lot of the problems, made a lot of mistakes. And so now we can achieve up to 6 generations of wheat per year, and we apply it wherever possible in our program for research and pre-breeding and working closely with some companies. And we apply it to both wheat and barley because I work on both. So to give you an idea, Dr. Norman Borlaug's favorite equation, apparently, genetic gain over time, but essentially you have selection intensity, selection accuracy, genetic variance, these components in that equation, and of course very important is years per cycle. And so this is exactly where this speed breeding technology, rapid generation advance fits in, and so it's about reducing the years per cycle. And that's exactly what Dr. Norman Borlaug was targeting when he developed shuttle breeding system in Mexico. So every breeding program on this planet is different and the way that they breed. But just to give you an idea of where this technology fits in, it's the line development phase, if we break it down. This field testing phase and the journey towards variety release, these components of a breeding program are often fixed in terms of time, because a minimum number of environments and years are required to evaluate that test material to make sure that that material is suitable for a farmer to grow, in terms of it being confident that it's adapted and has the right quality, etc. So it's this line development phase, and in theory, if we can go twice as fast in that phase, we can potentially double the rate of genetic gain. So that's the idea. So I had a quick, I could talk a lot about the speed breeding system, and I thought, well, what better way than to actually take you guys there. Welcome to the speed breeding system. This is just one of the glass houses we have here at the University of Queensland, fitted with lights to extend the photo period. In this glass house we can control the temperature plus or minus 1 degree, so it gives us great precision to manipulate plant growth. Of course, we often change the photo period from constant light to accelerate plant development, sometimes to diurnal photo periods depending on what we're doing. In this glass house, we have about 4,000 F2 plants. It's all trying to develop recombinant inbred mapping populations to map new resistance genes to rust. So you can see all around me the diversity in terms of plant height, maturity. So these plants are growing super fast. We're growing these plants in 100 cell trays. Now, it's 10 by 10 cells, and you can see when we grow them in this system, they generally produce one spike, one tiller each, and that's enough for the purpose, particularly for a single seed descent when we only want one seed. Now, often we grow the wheat or barley plants in pots like these. They're called ANOVA pots, and they've got one small hole in the bottom, and it prevents the roots from coming out. Now, the media that we use, this is actually pine bark mix, and we add Osmocote when we're potting up. That's a slow release fertilizer, but we also add liquid fertilizer, as if we don't do this, we have a lot of problems with nutrient deficiencies because the plants are growing so fast. So they really need those nutrients readily available. Otherwise, we run into problems like copper, calcium and iron deficiencies. So now you've seen where all the magic happens, but it's actually midnight here, so it's time for me to get some rest. Not for the wheat plants though, they'll keep growing. I'll catch you later. So just to give you an idea, so these are some of the plants when we grow them in pots. They generally produce about 3 or 4 tillers when we grow them, 4 plants per about a 2 litre pot. And you can see here this is a Suntop spike. Suntop's a widely grown variety in the east coast of Australia, and we can harvest the grain about this stage. This is about 11 days post-enthesis, but generally as a rule of thumb it's safe to harvest them about 2 weeks post-enthesis. So they're very green. They're underfilled, but what we do is we apply a cold treatment to get them to germinate. So we actually dry them down in the oven after 2 weeks after enthesis. We dry them for about 7 days at about 35 degrees. I find what works the best is we start off at about 30 degrees and then for a couple of days and then increase to 35. It's just a bit of a slower drying process. It's not as harsh. And then we imbibe them overnight, place them in the fridge for 4 days at 4 degrees Celsius, and hey presto, they all germinate. But obviously, things are different when you have grain dormancy in your cross. So what it does is it amplifies grain dormancy, this green harvest. So generally, you have to have a lot longer period in the fridge to get them, to make sure they all germinate. And sometimes with genotypes that are super dormant, we have to add GA as well to get them to germinate. So, yep. I guess to dive a little bit more into the details about what we use and how we do it. We get most of our pots and equipment from this company in Australia. I'm not a shareholder of the company, but if you're just interested for looking at the sort of stuff, and I'm sure you can find, us another supplier or another company in your home country or whatever. So Garden City Plastics, and they have all these Anova pots. I'm sure it's just the way we have the setup in our glass house. These work well, but I'm sure you can work out, different watering systems and pots that you can use. Again, so we use these, we have often grow plants in cell trays, so at high density. So when we do that, we grow the plants at it's up to about 1,000 plants per square meter. So we use these trays, 100 by 100 cell trays, like I showed you in the video, and generally for single seed descent or integrated phenotypic screening, so which I'll talk a bit about. Another thing that we do is kind of exciting is we use these sort of tubes, and so we do disease screening and we on seedlings, and we can make selections and transplant them into the field and all these sort of crazy things. But you can actually use like a vegetable planter to plant these things into the field as well. So it's really quite efficient then. So that's kind of cool. In terms of lighting, of course we're quite lucky at UQ that we don't pay the electricity bill. So we get away with having these elaborate high pressure sodium vapor lamps, which suck up a lot of power. So I guess if you were running a commercial breeding program, you just probably couldn't afford to have something like this. And so these glass houses we have also control the temperature plus or minus 1 degree. So obviously air conditioning is pretty expensive too. So anything to do with sucking up a lot of power. So that becomes your number one factor for, do you do this or not? And so, you have to think about low cost solutions. And so there's obviously been the technology behind lighting is evolving very fast. And there's a lot of reasons for that, and we might have the marijuana industry to thank for that. But this LED lighting has a lot of. We've played with it a bit. It's very promising. People are using it now. It's cheap, running costs. So, you'd be thinking about setting this up if you were going to do this at a large scale. Other low-cost solutions is we actually have the glasshouse facilities at UQ are not that good. So we have to share with a lot of other people working on sunflowers and whatever. And so the glasshouses we have are pretty dodgy, sort of like this, not that bad. But, I'm exaggerating. But what we did is we went to Bunnings. It's like a hardware store, and so we bought these spotlights, LED spotlights. They cost like about $60 Australian, and we put them up in the corners of each of the rooms. And so this can actually accelerate plant development. So existing glasshouses that you might have you could just set up a couple of spotlights like this, and it could help accelerate that generation time. So yeah. So also another option is. We've been thinking for a few years now that these shipping containers, that there's a surplus of them. They're everywhere. People just want to get rid of them often, and so you can buy them at a cheap price. You can buy them for about $3,000 Australian, and they're actually fully insulated. You can buy them with fully working air conditioning as well for a little bit more. And we actually, in Queensland, the state government have actually set up a couple where they have these fitted out with lighting, and they grow plants in them, and they grow wheat plants, and it's a low-cost solution to setting up a speed breeding facility. So you could connect several of them together, and so people are doing lots of crazy things with them. They're actually living in them. You can imagine if you set up one of these next to your portable speed breeding system, you could be pretty close to your work. Then you wouldn't have all the travel time.
What's the size?
You can get all different sizes, Mark. I'm not in the shipping industry.
4 by 20, or 4 by 4 by 40.
Yeah, in foot. Feet?
No, meter.
Okay, they measure
You can get the tall and the short version, Mark.
Yeah. You get long ones and short ones. And again, I'm not in the shipping industry. I was selling these things. I'm just highlighting opportunities. And actually, some companies have caught on to this. So this was one of the companies, I think, that have started in the US, and there's a number of them popping up. And this is all about, so I think some of them are actually growing like vegetables. It's about, I think Kathy actually might, I think some of the Gates people actually made me aware of this. And so yeah, it's maybe, I think, for growing vegetables in cities and stuff like that. So I think there's a, anyway. So what we thought was, we have this system, we can go fast, we can go through generations quickly, but wouldn't it be great if we could select for some desirable traits? And so if we did that, if we look at the genetic gain equation, what are we targeting? We'd start applying selection. So we're targeting selection intensity and selection accuracy. And so I won't talk much about this because Cecile Richard is going to tell you a lot more details about some of the exciting screening methodologies we've developed. So for grain dormancy, rust resistance, and some exciting stuff on root traits and yield component traits. So I'll leave that to her. But in a nutshell, when we first started this work, we thought there would be a lot of benefits to evaluating some of these traits that have high heritability in this system compared to the field. We know measuring some of these traits are difficult sometimes in the field, and if we could actually pull it off, it would be great if we could couple it with that rapid generation advance and cycling system to enrich breeding populations. So Cecile will talk more about this, but we developed a system for a triple rust screen, so all 3 rust pathogens in the 1 plant generation. And so we coupled that with a screen that we developed, or assay we developed for grain dormancy. And we applied these phenotyping techniques in combination with the rapid generation advance through speed breeding, some crossing schemes to stack multiple genes, and high throughput DNA markers to maximize genetic diversity through the procedure. And so this is the sort of example. This is an example of what we did, and this is achieved in about 18 months. We had about 7 generations. We generated back cross-derived introgression lines, and an interesting step here was that we screened very large segregating populations using this multi-trait screening approach, and on the same plant generation, we were able to back-cross those selected individuals. So those individuals that have that special gene combination, we're able to back-cross them the same plant generation simply by chopping them back. They'd already flowered, but it didn't matter. Chop them back, transplant them into pots, add some fertilizer, lower the temp, they retiller, they grow, we cross them. And so that was important because if you go to the next generation, they all segregate again, and then you're crossing non-dormant and rust-susceptible genotypes. So in the next generation, we actually did a intercross. So we intercrossed those back-crossed plants, and combining those genes produced another big segregating population, and repeated this multi-trait screening process. So then it's important to get to the field. So after we did that and increased seed, there's a number of companies in Australia commercializing this material. And so we've developed a lot of different germplasm through this pipeline, but just a couple of case studies. This one we worked on was wild catchum, widely grown through West Australia, but became susceptible to rust, and growers moved away from it. And so we worked with this background, produced these introgression lines, and not only did they have the rust traits, but we'd actually improved yield in a number of them, so compared to the Czech wild catchum. And interestingly, in that time, while we were working on this material, and wild catchum is quite an old variety, maize was released, and so a number of the lines that we developed were equivalent yielding to maize as well. So I guess we're in the position that we are public breeders and researchers, and so we can only work on populations that are detached, I guess, from the real world in terms of breeding. And so we did this work on it as a case study to highlight what could be done using this sort of approach. And so ideally, you want to be working on this sort of thing for elite breeding material, and so you're working on the best and the most elite material. So I guess imagine what you could do if you're combining top crosses in this way. And another example, this was at a field day where we had over 300 farmers, and so I guess this is a different variety suited to Victoria and New South Wales, and I guess it's kind of a nice feeling to see something that you've developed in this system in the field and yielding well. So to give you an idea of how we have optimized the selection pipeline that we adopt, and I guess it's important to say that most or majority of the breeding companies in Australia at the moment are using speed breeding to some extent. A number of companies do work with us, and so this is an example of what we might do for any particular cross. We can screen for grain dormancy. It's easy to do. It's a grain trait. We can do big trays germination, select the dormant ones, grow them on in the speed breeding system, screen for yellow spot. Great, easy trait to deal with in terms of being at the seedling stage. Grow them under the speed breeding conditions to accelerate plant development. Get those adult plants very quickly. Screen for adult plant resistance to the 3 rust pathogens. Reselect for plant height and maturity because we know these things are correlated with the field. And then we have this highly selected set of individuals that have these great combination of genes for the target traits. And so we can do anything with those. We can proceed with single seed descent and repeat the selection process the next generation to help fix the genes, or we can back cross, intercross, or we can couple it with a marker program and actually apply marker assisted selection for known genes. So when we first started this work, we were pretty crazy and we're doing all these crazy things, and it wasn't until we started working closely with breeding companies that we actually realized the importance of adapting it to a breeding program. And so you actually have to back off the pace a bit and actually redesign a breeding program and work out where it fits. So it's not about doing 6 generations per year. It might be about doing an extra 1 or 2. And so it's essential that it fits with that field testing scheme. So this is not about forgetting about what happens in the field. It's a tool that has to be married up with everything else that we're talking about here today. And so a lot of the companies, it is about redesigning the breeding program, rethinking with all these technologies. It's not just speed breeding, it's genomic selection. It's how do we integrate it, because you have to change what you're doing. And that's why it's really challenging. So to give you an idea of some of the exciting stuff we're sort of starting to do now with Jessica, we're starting to combine speed breeding with genomic selection. And I guess this figure gives you an idea of how it might work. And in theory, if you're going twice as fast in terms of your population development, you could double the rate of genetic gain from genomic selection pretty easily. Obviously, it's a lot more complicated than that because you have to rethink how often you retrain your population, how do you keep up with going so fast. So there's a lot of challenges there as well. Speed breeding works in other crops, which is pretty cool. I know some of you don't work on, this is a rice breeder here. So it works on barley, well, of course, we apply it at UQ. Peanuts, a peanut company in Australia have developed a system for peanuts, and they run their whole breeding program through the speed breeding system. So it enables them to achieve 3 generations a year. So that's pretty cool. And we're testing some systems for sorghum at UQ with David Jordan. So yeah, just to show you that some pictures of canola, it works. I don't know, actually, Dave was helping us with that, and I don't know much about canola, but it grows fast. So that's cool. So at the end of the day, speed breeding can be used to rapidly improve existing cereal cultivars. So ones that are out there widely grown by farmers that might have deficiencies. Gene pyramiding and top crosses, so elite by elite, quickly combining those characteristics. And rapid integration of novel genes from land races, so for research and pre-breeding. So it's something that was really time consuming, and a lot of people just didn't do it because it wasn't worthwhile, it just took so long. Now we can maybe resurrect some of those old genes and bring them into modern wheats very quickly. So just want to acknowledge GRDC here for funding a lot of my projects. My colleagues, my fabulous PhD students, I couldn't do anything without them. And of course, Dr. Mark Dieters and Dr. Ian DeLacy for concepts relating to speed breeding at the very early stages. So yeah, that's it. So some questions.